talk about proteins and lipids. So, starting off with proteins. Proteins are made of organic molecules, again, that have carbon, hydrogen, oxygen, and nitrogen. These are called amino acids. So, proteins are made of amino acids. What do proteins do? Proteins can make up structural components in animal cells, such as skin, hair, muscles. They're also functional. Enzymes are a class of proteins. Hormones are a class of proteins. So they're functional in keeping us going as well as structural. So let's talk about amino acids that make up proteins. They're an organic molecule. This is generally what they look like. And you hook amino acids together to build proteins. The basic structure, they do look like this. There are 20 different amino acids. They all have the same basic shape here. The thing that's different is called the R group, which is circled in blue here. So, what happens when we link amino acids together? Before we go through the slide, you now know that amino acids are linked together in ribosomes. Pretty exciting, huh? So, in the ribosome, a peptide bond is formed between two amino acids. That's a fun word, peptide, don't you think? So we've bonded a few amino acids together with peptide bonds. It then becomes a polypeptide. A polypeptide is just another name for a long chain of amino acids. So when does it become a protein? It becomes a protein when the polypeptide chains are folded into their proper shape. So this folding can happen right in the cytoplasm as they exit the ribosome. This folding can happen in the endoplasmic reticulum, or a little bit of folding can happen in the Golgi apparatus before it is shipped off to another part of the body. So how do you test for proteins? You're going to add the biuret solution to the food and swirl it around, and that's it. If there's a protein present, then the food mixture will turn a violet color, and it's generally a light purple in color. Now, moving on, let's talk about lipids. There are several kinds. Here they are: fatty acids, triglycerides, phospholipids, steroids, waxes, and pigments. These three things that we're going to talk about now are common to all of these lipids. They don't dissolve in water. So oil and water don't mix. We've got oil is actually composed of lipids, and we know that it doesn't dissolve in water. Lipids have a higher ratio of carbon and hydrogen atoms than the other macromolecules, and they are great at energy storage. So lipids is another term for fats, and we all know that people are great at storing energy as fat. So fatty acids, which is a classic lipid, is a long carbon chain. Look at all these carbons. And it's attached to a carboxyl group. Remember the functional groups from previous notes? So we have our carboxyl group, which is called the head, and then a long carbon chain, which is called the tail. Now I've got two different fatty acid chains here. One of them, the hydrogens, completely fill up all the extra space on the carbons. Here we have some that are missing. That's the difference between saturated and unsaturated fats. Saturated fats, there's no extra space on this carbon chain. An unsaturated fat is missing some hydrogens. Now what this does is this causes the fatty acid to bend. So the link that it would have formed here and here will now form together, and this will cause the chain to bend. Triglycerides, three glycerides. So it's a molecule made of 3 fatty acids joined together. It can be saturated or unsaturated. Triglycerides are generally, if they're saturated fats, they're found in animal products like meat, butter, and cheese. Unsaturated fats are found in plant products like seeds and nuts. Lipids can also be structural in nature. Phospholipids are an example of a structural lipid, and phospholipids are made of a glycerol molecule, which is the head, and then 2 fatty acid tails. Their function, these guys make up the cell membrane. And we'll talk about the cell membrane more later. But the fatty acid tails all point towards the center of the membrane, with their glycerol heads facing the cytoplasm and then the environment outside of the cell. How you test for lipids. You're going to add Sudan 3. Sometimes the test may say Sudan 2 or simply Sudan. It all works the same. You swirl it gently. If there are fats present, they will turn red. In our test, they sometimes turn a pinkish color. You'll just have to read the chart. I would say if it's any sort of reddish color is indicated, that fats are present. We're going to take a moment and talk about nucleic acids just briefly because they are one of the macromolecules. First of all, we don't have a test for them, but nucleic acids are DNA and RNA. A monomer of a nucleic acid is the nucleotide, which is made of the nitrogen base, a sugar, and a phosphate group. So here's what you need to be able to know.